Thanks, Claudia. So let's see here. Can we advance the slides? Great, thanks. So when I thought about this title, the need to expand scope, life beyond probes, I struggled with kind of what angle to pitch, and there's actually many angles. So I'll talk about a couple of them. At the Gates Foundation, we've now instituted a process that we call product development workflow, and it very much follows standard product development, regulated diagnostics development in a company. And starts out, you have a research concept, and if that looks promising, then you go on to different phases. And if I had to simply distill what happens at the end of each of those phases is, at the first one, you want to answer the question, do we want to do this? So you got a technology platform, looks promising, or you got a biomarker that looks promising. Okay, how much do you want to bet on this working? So you got to answer this question. So we have like an exit phase gate question about, that basically says, do we want to do this? If you go into that next phase, phase one, where you're now putting serious money into this, you want to really do your diligence on feasibility and planning. At the end of this, you should be able to identify your risks, have mitigation strategies, and know pretty much what you're getting into. And to be able to exit that phase, you want to answer the question, are you sure you can do this? Because it's going to get more expensive as you go further down the pike, right? If you get into the next phase, what we call phase two, or design development, this is now really heavy into getting to a final device which you would lock down. And it's at this phase that you do analytical validation, and you have a really good predictor whether this thing is going to work in a clinical setting or not. So at the end of that, to get out of that phase, you want to be able to say, yeah, we can do this. We're really sure we can do this, because we have evidence, at least from an analytical perspective, that the device works. So now you get into your clinical studies, and you demonstrate clinical validity and or clinical utility, and you really get a claim. There's an intended use statement. It is it's approved or cleared by regulatory agencies, and the development team can at least say, okay, we pulled it off, we did it. I was talking to a colleague at the break, and we were talking about development or technological development versus implementation, and it's kind of like an iceberg. The part above the water is the technology side, and the part below water is the delivery side, the what then has to happen to make it real and implemented. And the question there is that can we do this on a sustained basis, that this is going to actually fly, there will be uptake and there will be impact. So this is kind of a simple stage gate process that we will apply for virtually all of our grantees that are going into product development. Now, along with that, we've got some deliverables. And these are not going to be a surprise to people that are in this business, but for each of these stages, across multiple functions, whether it's program management or marketing, legal research, development, manufacturing, regulatory, et cetera, there are certain things that we're going to ask to be articulated and developed over the development pathway. And this really forces an early conversation between us and the grantee about how exactly do you envision this to look like at the end of the day. So we coined this phrase, end-to-end thinking, from the very earliest research thinking all the way through implementation, uptake, and impact. So, and we'll have phase gates where we have go, no go. So there are going to be clear milestones, and this will be a transparent conversation with the grantee in the beginning. But this is kind of our end-to-end thinking, and it's very much the devil's in the detail. Now, one aspect of going beyond probes is let's go beyond the technology, the perfect widget. Let's go into a broader realization of a patient and the pathway that a patient encounters in looking for healthcare. So this is a kind of a map that just walks you through a patient who starts to seek care. Is it available? Or if they find a care center, were the diagnostic tests there? Did the supply chain work? If the supply chain worked, was the product still shelf life viable? You think about, in order to go through this pathway, you have to order the right tests, and maybe the problem is a healthcare worker doesn't know which test to order, or the tests don't exist yet, or they're too expensive, even though you might have lots of publications on cost effectiveness. If it's cost effective and can't be afforded by the budget, then you're not going, that chain getting to A to B, you've broken one of the links, right? So then, let's say that the right diagnosis was done, the tests were done, and now the question is, are the drugs available? Okay, and if the drugs are available, is the patient initiated on treatment? And if the patient initiated treatment, did they finish treatment? Were they compliant? And only when you get to the end of this patient pathway and everything has to click, right? Those links have to be established. Then you get to a place where you want to be, and this is a point of success from a patient perspective, and they're cured. So this is one thing that, as I look at, diagnostic technology innovations, this patient pathway leads you to another perspective, a holistic perspective, and I plagiarize this from Find, I think, and Madhu got it from you, but this is a really nice holistic view, is that you've got a patient who is symptomatic, okay? If it were possible in a highly decentralized setting to have a home pregnancy type technology test that could have really high sensitivity and okay specificity, and you could use it as a rule out. Therefore, you're saving that country's budget a lot of money. If this were an inexpensive test, then you don't have to bother that person to go to a microscopy center to get a more expensive molecular test. So this is a need that is articulated in one of the TPPs that WHO, Claudia talked about a minute ago. Then, of course, we've got this state-of-the-art rapid molecular and DST innovation. And GeneXpert really, Cepheid, many years ago, a friend of mine at the FDA said, there's a saying at the FDA, instead of the early bird getting the worm, the early bird gets the snake. And what this means is Cepheid was stepping into a bold territory that nobody had gone before. And the problems that we all went through, Cepheid included, right? As Claudia said, we all went through an enormous learning curve, right? So this is really, really valuable, and it's really gratifying to see now that the uptake of GeneXpert is on an incline. Well, beyond case detection, whether or not you have upfront DST or not, there's a need for culture, and sequencing, no doubt, will come and play a role in this. I'll talk about that in a minute. But beyond that, we've got the need for shorter drug regimens. So the last couple of days, we're talking about new regimens coming that could potentially shorten treatment times. Then is the patient are they compliant? So adherence monitoring is an unmet need. And then electronic notification and tracking, medical records, and all of this, what is connected, there's a backbone to all this called electronic TB, Information Communications Technology System, so an ICT. And Find has really led the way in trying to get a conversation to standardize this so that any device could, by rules of communication through application programmable interfaces, could connect with a primary care health system. So this is one way of going beyond probes. Now, next generation sequencing is going to be really interesting to watch, I think, going forward. And there are lots of publications that are coming out on this. And, it's just got the potential for a lot of problems that it can solve. However, we've got a ways to go. We have a very important initiative that was discussed yesterday called the RESEC TB database. And I think I've got a slide on this. Yeah. So this is one of those things that, why someone didn't think about this earlier is beyond me. But, to a large degree, research that happens in healthcare is very much siloed, because a lot of the research that we have going on is funded by grants, and there's a constant battle to get grants. And so principal investigators are successful in their academic positions because they can get grants. And there's not a lot of incentive to share, although you do see it, but largely it's driven by my grants, my program, and this happens all over the place. So this is true with TB databases. You just look at all the colleagues in CPTR and find that have been looking into this. It's like, how do you get your head around this? And so we made the decision to try to develop a global standard, universally harmonized, accessible database that could be kind of the one-stop shop to help, in the first phase, understand what is the molecular basis for drug resistance. Let's dive deep into that with a lot of power. So there's a real need for those of you that are working in the sequence arena to help us populate this with quality data, whole genome sequences, so that we can begin to mine this to identify, in more explicit detail, the molecular mechanism behind drug resistance. For some of the targets, they're well established, and some of the targets, they're not. So next-gen sequencing is going to play a role in this. And I looked at PubMed publications for the first 15 years of PCR and looked at the ramp-up. And that was from 1985 to 2000. And then I looked at the past 15 years searching for next-generation sequencing. And not quite as steep as PCR, but it's a very similar profile, because the applicability of next-generation sequencing is so enormous that there's a lot of interest and research going on in this. So we think that this ReSeq TB database and next-generation sequencing is going to allow IVD manufacturers who are going to be using probe-based technologies to better design their assays. That is the first goal of this program. Having said that, and Angela will talk about this, talked about it a little bit yesterday, and it'll come up again, I think, and that is that what does this look like in a generation? And I was talking to some friends yesterday at lunch about, remember there's a movie in the mid-90s called Gattaca. Anybody remember that? Okay, there's a scene where this woman walks up to a window and she gave a hair follicle or something for her boyfriend and said, I want this DNA analyzed. So 5 minutes later or something, she gets this scroll with this massive sequence and she's checking them out, right? Well, it's probably not going to be like that, but in a generation, we are going to be stunned at what we see. I think this sequencing is going to become more and more prominent. It will happen first, of course, in the developed world. The question that we are grappling with at the foundation is, should we try to catalyze its emergence in the developing world? And amongst the many bets that we would place, this is one topic of conversation. So for next generation, I think you can say that it's enormously attractive for multiple purposes, but there's a lot of challenges. 10 years ago, if we were talking about the human genome for under $1,000, people were going, yeah, right, give me a break. Already, crossing that barrier. But despite this attractiveness, there's a lot to do, right? So there's going to have to be an enormous collaboration. Silos are going to have to come down, and we're going to have to figure out somehow to collectively pull this off. And I think, it was interesting. Joe Biden, Vice President of the United States, was in Seattle about a month ago, talking about the Moonshot Cancer funding initiative. And one of the researchers at University of Washington who hosted him was talking. This is a news press, news segment, and he was saying, "Yeah, we need more funding." And Joe Biden said, "Yeah, absolutely agree." And then this guy said something that I thought was quite remarkable. Please help us break down our silos. If we are going to really tackle some of these intractable problems, we cannot continue to do, our own thing. That will still happen, but to the degree that we can come together. Clinicians, regulatory. Yesterday, Heike talked about, the problems with sequencing. And it's so powerful and so flexible. How do you develop regulatory policy on that? Well, it's great to see that conversation is starting. So for us, the next step with looking at next-gen sequencing is the technology doable? Okay, that question has already been answered. Can the technology be distilled down to a microscopy center in Mumbai? I don't know. That's another issue. But for us, this part of this end-to-end thinking is that we have to assess the business case. If the widget works, and you haven't solved the back-end problem of delivery and implementation and uptake, then you've broken a link. You don't get to point B. You don't get to the final. So this first question in the early phase gate is, do we want to do it? So one of the deliverables is to develop a business case and try to answer the questions up front. It's very much an extension of what our speakers, Dan and Palau, said early on, that is, can we do this in a smarter way to more likely ensure success? So with that, I think that is my last slide. Thanks very much.
Thank you, Jim. Any quick questions to Jim before we move on? If not, maybe I actually have one quick one, Jim. You have put up the pipeline here for the Gates Foundation, and what I think would be interesting to briefly highlight how you at the Gates Foundation decide what to invest in, and if that is maybe just a small subset of what the global health community has defined as priority needs, or maybe even totally outside of those priority needs. So if we take the TPPs that were defined, how you decide that the focus would be now on triage and not, let's say, on DST, or if you could just quickly highlight that.
No, it's a good point. So the first thing that the TB program strategy team at the foundation, which is the team in charge of virtually all the investments that we do, whether it's drugs or vaccines or diagnostics or delivery, in diagnostics is guided by those 4 TPPs. So the question that we grapple with is, what weight do we ascribe to any of those 4? Which would give you the greatest ROI? And a colleague at the foundation, Dan Hartman, he puts it this way: how can we do the most good in the shortest amount of time with the fewest resources? And it's really quite interesting. You think, well, Gates Foundation, resources, it's not a problem, right? But we actually, we must be stewards of the funds that Bill and Melinda have provided us. So there's a lot of thinking that goes into where do we place our bets. And the good thing about the Gates Foundation is it's more risk tolerant than most companies who answer to Wall Street on a quarterly basis, right? So we can place bets that are more risky. When we look at the 4 TPPs, there's clearly it's a legitimate need for all of them. And we had hoped that funds would be available to pursue each of those, because those are the prioritized TPPs. But right now we're in the midst of a strategy refresh, and we're asking those questions. What weight would we ascribe to any of the 4 of these? And so the answer, the jury is still out, Claudia, so you can't answer it. I think the next generation sequencing, we'll talk about it later, but I think there's going to be a need for a TPP to be developed for that.
Thank you very much. Okay. No other questions that popped up?
Maybe I
one
should
quick
ask,
one.
but in terms of breaking down the silos, what's the sort of strategy that will force scientists to contribute their data? Does it make it a collaboration? Do you want to make it that there's a policy that you can't publish without putting it into these databases?
Yeah.
What's the strategy?
Yeah, it's a great question, Rob. So at the end of the day, so we have a project coming up, and we've concluded that if you have an all-in-one cartridge like GeneXpert, like the MTB RIF cartridge that does sample prep and AMP detect in the back end, the cost of goods of that will preclude reaching price points that we want to get to. So we're going to pull out sample prep. I'm going to issue a targeted RFP for sample prep. And we're pretty convinced that there's no single institution on the planet that has all the competencies to pull this off. Even the large diagnostic companies like Abbott and Roche don't know these markets. So there's a real need to form consortia. How do we do this? So we clearly have to answer the needs of all the stakeholders to pull this off. And for the academics, I think clearly, the small companies and the academics are where innovation happens. The large companies are where delivery and execution happens. Those have to come together. So to answer your question, we're still on a journey to figure this out. We recognize that we have to address the needs of everybody to pull this off. But it has to be done in a way where there is a mutual recognition that it's the consortium that wins, and not the principal investigator who happens to be the first guy on the grant application. So good point, and we're still on that journey.
Thank you very much, and thank you for the